All right, next section. So this is a little tiny section, but it's got some interesting stuff in it. So let's go. So we've done the innate immune response. Now we're doing another piece of the innate immune response, but with a little twist on it. So this is called the collaborative cross, and this is a very powerful technique that's done. It's very expensive to do, but it can get you an answer, a genetic answer, pretty quickly and very powerfully. So the idea here is that they started out with a bunch of mice, and so this is something you can do in mice. And they start out with 8 different kinds of mice, and so they give them each a different little color on here. And then they crossed these mice. They crossed peach with yellow and green with blue. And so they made different groups, and then they took the males from this group and the females from this group, and boom, crossed them again. The same thing here, etc. And they just go through all of these possible changes, and you end up with a series of experimental lines that mix all the same genes. And so this will then let you figure out genetic susceptibility. So you'll test out, first of all, in all of the regular mice, you can test out your virus. And so let's say it kills the yellow one and does not kill any of the others. You can say, oh, the yellow one has the gene somewhere, but genome's a big place. So then you can go through the last generation. So they're only showing one here, but really this tree is broadening out because they're making every possible combination. And so in the end, they've got something like 100 different lines of mouse. And these things have a known, so they've already mapped out their chromosomes and they know where each chunk came from. And so if you say, well, they've got something from the yellow mouse, the yellow starting mouse, you can then try it on all 100 of these last generation mice and see which ones look like the yellow mouse and which ones don't. Then you can narrow it down. And so maybe all the ones that have chromosomes 1 through 13 and 15 through X and Y do not have this. You can say, well, then it must be right here in chromosome 14, and the only part that comes from the yellow is right there, and boom. That fast, you can actually get down to an exact little locus that'll give you maybe 4 or 5 genes, and then that really narrows down the process so you can go in and figure out which of the genes is responsible for the effect that you see, or if it's a combination of all the genes. So the experiment uses a couple of mouse lines, and they are they start out from 8 and then get scrambled up in neat different ways. Okay, so this is like big science, takes a lot of money. It actually costs something like about $3 per mouse per day to house a mouse. Plus, some mice are quite expensive. You would normally buy them in rather than breeding them yourself, because when you buy them in, they will actually do the genetic tests and make sure they are what you think they are. And if you try and grow your own, sometimes they lose the mutations that you bought them for in the first place. So these are some collaborative cross mice, and so we've got resistant ones and susceptible ones. You see, they both have a similar sort of loss of body weight, so they both get kind of sick. But one of them bounces back and the other one doesn't. And the other one doesn't because the other ones all died. Right here, bam. 100% death by day 6. And so they weren't able to weigh them on day 6 because they were already dead. They'd lost too much fluid by that point. So yeah. Pretty severe. And in the resistant ones, they had 100% survival. So now all you've got to do is do this in the entire panel of collaborative cross mice, and then you can start to narrow it down to either a region of the genome or, in some cases, a single gene. And so here are the differences in the pathology. So, all right. At day 5, susceptible mice, which are in panel C and D. So resistant mice are resistant are in E and F, And susceptible are in C and D. There we go. C and D, susceptible, E and F, resistant. So what you can see here is that this is the normal color of a liver that still has all the blood inside of it. And here is the liver of the Ebola infected mouse. And you can see that, and this only works because Ebola does infect mice. It can get into their macrophages. See, it's a little darker, sort of coffee color, as they say. That's because you've got some hemorrhage going on in the liver. So that's a hemorrhagic hepatitis. And then over here, you can see the spleen is not even really poking out. It's maybe that little tip right down there. It sort of hides back behind the liver. And here in this case, you can see the spleen is sticking out, and it's highly, highly swollen. It looks like it's about to burst. Yeah. So, yeah, that tells you that the virus is damaging both the liver and the spleen in the mice, just like it does in the people. And so with an animal model, you want it to match as well as possible with what happens in humans. You want the same sort of disease with the same sort of mechanism. Otherwise, it is not a good animal model. You don't kill mice just to see what happens. You only do these experiments if you're going to gain something that's going to be useful for treating people. That's the only ethical way to do it. And so with the collaborative cross, here's what they found. They narrowed it down to a chunk of the genome that had these genes on it. So you got your serpins here, and you got a bunch of other little things there. And so they then went in and looked at those with a thing called transcriptomics to see, are these genes being turned on more than usual or less than usual in infection? And so in some of these cases, like the blue means turned off and the red means turned on. So here's the same gene at 1 hour or 1 day, and it starts off here, but it starts out on in these susceptible ones. And so it looks like early and sustained expression of serpin C1 may lead to susceptibility. So something like that. And if there were no differences in between the two, then you could say, well, it's probably not this one, it's probably something else. Anyway, with this combination of transcription profiling, so looking at what RNA cells were making when they were infected or not infected, and looking at the collaborative cross to see which mice were susceptible and which ones were just totally Ebola resistant, they narrowed it down and found out that it was the Ti and Tec proteins that seemed to be the big determinant of whether mice live or die from Ebola. And of course, once they figured this out, they went and tested it out in human cells, and it turns out to be absolutely the case. Ti and Tec are also very much important in human Ebola virus infection. Okay, so here we go. Here's the Ti and Tec pathway. And so Ti1 and Tec are very similar proteins. That's why they've drawn them similarly here. And so Tec can bind to Ti1, or two Tecs can bind together, either way. And they are going to be activated by angiopoietin. And so here's this ANGPT1 and ANGPT2. So angiopoietin 1 is going to activate Tec, and angiopoietin 2 is going to inhibit Tec, and Ti is going to inhibit angiopoietin 1. So if you're into circuit diagrams, what you've got here is a nice sort of feedback system. So let's say angiopoietin 1 is activated. It's going to turn on Tec. Tec will have its effects. Great. Your signal will get through. Let's say you don't want that signal to get through. You'll turn on angiopoietin 2, and so that will stop Tec from being activated, which will cause no signal to come through at this end. But let's say you've already made some angiopoietin type 1 and you want to get rid of it. Well, you just have that cell make Ti1, which then blocks angiopoietin 1, so it can't get to Tec. It just binds it. And so you have no signal coming through. So you have multiple ways to get a signal or not get a signal. And there's all these other arrows that are in here are all connecting up. And these are also, different ways of signaling related to these proteins. So the idea is that activated Tec is going to activate NF, lowercase k, so kappa, NF kappa B. And you remember that one probably from our little page with the immune response to RIG-I and MDA5. Remember, we were trying to make interferon response factor 3, IRF3, and NF-kappa B, because these were two things that were going to make an inflammatory response and were going to protect the cell. A nice little interferon response. So when I say inflammatory response, that would be, I'm going to use that as if it's the same thing as an interferon response. So interferon response just being a specific type of inflammatory response. So yeah, we don't fully understand the function of Ti1, but it's real similar to TEC, so it probably is very similar. And they're both tyrosine kinases, and so they are going to add phosphates to each of these little factors that are then going to go out and activate something, which will go into the nucleus and bind to DNA and start transcription. So the TEC protein was identified with the collaborative cross, and that's what it does. And that's the end of this little mini section. Now we're going to go for the big section at the end.